Okay, so we have the end of the program time, save for 10 minutes where we're kind of have some final closing remarks, where we have 68 persons on the call right now. And instead of breaking out into individual sessions with perhaps groups of 15 or 20 persons or so, we can actually stay within the larger group and then manage a nice discussion breakout session with us all. So where I thought we would start is I will share my screen. Some topics have been aggregated that have been discussed today. And I would like to do a vote, a show of hands, to kind of get an assessment of where our interests lie, where we can maybe start the discussions. And actually, I would invite anybody who wants to turn their camera on and feels like it won't damper their wireless connection to go ahead and turn their cameras on so we could see our faces together. And I'm going to now share my screen so we can now vote on some topics to begin a lively interactive discussion. Okay, so right now we have 7 topics and we're going to focus on polling which ones might capture the most interest to start off with. So everybody might be familiar with the hand function in Zoom. Yeah, so basically what you can do is use the hand function and I'm going to call out a topic. And if you have interest, raise that hand. Okay. Okay. So the first topic is using animal models to help study translational concepts. If you have interest in this topic, please raise your hand. Okay, we have 12 participants who raised their hands, 16, 18, numbers are going up, 20, 22.
I guess like you can raise hand to multiple topics, right?
Yeah, we're gonna be voting down these 7 topics.
So yeah,
right here.
Just don't be shy and then just raise your hands as many
I think
topics as.
I think that people who don't know how to raise their hand, they could perhaps write a message into the chat box.
Okay, excellent. So we have about a third of participants raising their hand. That's a fair amount of interest. Everybody lower your hands down. Now, one of the topics that was kind of hot and on the list yesterday was cancer. We discussed the prevalence of breast cancer. We also discussed brain cancer, tumors, tumor progressions, cancer drug discovery. If anybody has interest in that topic, please raise your hand. Okay. Okay, great. Hands lowered. And the next topic is based on botany, plant extract research. We have a lot of biologists on the call as well. So if you have the interest lying in plants and botany, just please raise your hand if you would like to go in that direction.
Could I quickly suggest that I think some people are having low internet connection, so they do not lower their hands, probably because of the disconnect from the time you say it to the time they heard it. So let's give them like a few extra seconds for them to lower their hand before you move forward.
Excellent idea. Okay. Okay. The next topic is a bit general. This could be generalized as well in discussions. It's about chemical analysis and analytical methods. So giving everybody ample time, please raise your hand if you have interest in this topic. Okay. Okay, so we have 3 other topics to vote on. If everybody could lower their hand. The next topic relates to neurodegenerative diseases, pathological proteins. You might remember some of the talks that touched upon those topics yesterday. Please raise your hand if you have interest in that topic. Okay. Okay, it looks like this topic and number 1 might be the 2 top hits so far. And okay, we have the 26 hands logged. Go ahead and lower them. And the next topic relates to ecology, evolution, evolutionary origins, and species-specific adaptations of the nervous system. If you have interest in this topic, please go ahead and raise your hand. Mm-hmm. Okay. And we can lower our hands when you're ready. Okay. The last topic, COVID-19, how it might relate to chemosenses, how it might relate to any other part of the brain, brain function, or just the prevalence of COVID-19 across the globe or in Nigeria. If you have interest in that, please go ahead and raise your hand. Mm-hmm. COVID-19 was lung cancer. Yeah. Okay. So based on that, and everybody can lower their hand, we have 3 hit topics. So we're going to kind of structure this as a discussion where rather than having myself or any of the organizers speak, to kind of encourage anybody to rise up, ask questions, and let's just start discussing together as a group. Perhaps we can start with using, well, you know what? I have to say, we had the most hands raised when it came to topic number 5. So I feel like this would be a great point to discuss with our breakout discussion round. So perhaps you want to write into the chat any particular thoughts, ideas, questions you have. And I could just kind of read them out and pose them to the group to initiate and start an interactive discussion with everybody. Perhaps I'll pose the first question. Is there anybody on the call who's actually studying or conducting research on any neurodegenerative diseases?
Really, can you repeat your question, please?
Yeah, my question pertains, and I'm asking the group as a whole, if actually there's anybody on the call who's currently conducting research on any of the neurodegenerative diseases.
Okay. Okay, so really there is some question. Could you read those questions? Perhaps a question from Khadija Abakara Bobo.
Okay, yeah, that's a great idea. And, you know, I also want to add that there are about 5 people who have raised their hands to say that they are currently conducting research. And those might be the people who would maybe best be able to lend their expertise and help answer these questions. So the question of mind is, can accumulated pathogenic protein of the brain migrate into other brain structure or organs that could result in another form of disorder? So, Salu, if you want, you could turn on your microphone if your connection allows. I see that you did answer that question in the chat. If you would like to elaborate, you can feel free to unmute yourself. Mahmoud, everybody has the capability to unmute themselves, right? It's not just the hosts.
Yeah, so I've given them the ability to talk.
Okay. Okay, great. So Salu, would you like to unmute yourself and elaborate on your answer?
Two people are unmuted. Philip, do you want to talk?
Can you repeat the question?
Yeah, if given time.
Go ahead, Philip.
Hello? Am I on? Hello?
Please go ahead.
Am I on? Hello?
Yes,
Please go ahead. We can hear you.
loud and clear.
Can you repeat the question? I can get you.
So actually you had answered in the chat that we have, for example, a few other, so the question was related to pathogenic protein accumulation. And then one of the answers that you had actually provided was relating to Alzheimer's disease, Parkinson's disorder, and memory disorders.
Okay, I'm currently working on plant extracts that have the ability to reverse neurodegeneration, especially that of Alzheimer's disease, as well as Parkinson's disease. And those that can ameliorate the memory dysfunctions. The plants were actually selected from an ethnomedicinal survey that was carried out in the southwestern part of Nigeria. So currently we are doing the acetylcholinesterase inhibiting activities, as well as the phytochemistry. Then some of them have been claimed to have anti-inflammatory activities. So that's what we are doing currently in the lab, with the hope that we are going to do the in vivo experimental models before we eventually do the mechanistic studies. Currently, I'm about entering into the aspect of the experimental in vivo with the use of animal models like Y maze, elevated plus maze, recognition task, and then water maze as well. That's currently what I'm doing. Thank you.
Thank you. So we have a comment by Royhan in the chat about systemic toxin injection models or intracranial injections. Which do you think is best for translative research close to clinical outcomes in neurodegenerative diseases? Would anybody like to answer that question? We have a hand raised by John Olin Realu. Would you like to answer that question perhaps?
Could you repeat your question, please?
Sure. So the question actually comes from Royhan Valerin. And
Yes.
what he wants to know is, what do you think might be the best for translational research, bridging to get closer to clinical outcomes in neurodegenerative diseases? Should we be looking at systemic toxin injection models or intracranial injections? What do you think about that?
By saying intracranial.
Can I make a comment?
Yes.
I know that the time will not be on our side. Instead of people sending us random questions that can be addressed maybe privately, why don't they ask us about how BioRTC can help them towards addressing those types of questions? So you see, we can then be able to think about how we can put those things in a medium, short, medium, and long-term strategy. Do you guys agree with that?
Yes. In terms of time, I think we can get right onto what is most directly related. And a lot of people agree in the chat. So is there anybody who would like to raise their hand and kind of try to draw the link about what we can do with BioRTC in terms of addressing neurodegenerative diseases, or potential collaborations with those other researchers conducting this line of research? I see Hassan Sani raised his hand. Would you like to speak?
John, you had your hands raised since, and I think you've been given the permission to talk as well as Philip. Can you unmute and talk, as well as Hassan?
Thank you very much. Taking from what Professor James Olopade mentioned yesterday, I know in Africa, especially in Nigeria, we have been well blessed with cultural diversity. And studies have shown that less than 5% have been, studies have been done on these plants. So with BioRTC, it will be a great advantage for us if we can exploit as much as possible by doing good science, as mentioned by Professor Olopade yesterday, such that I'll be able to have harness this opportunity of testing, evaluating these plants as much as we can, and even advance the research by collaborating with people outside the country. We can make use of BioRTC as a platform, and Professor Amadi has also suggested BioRTC should be made in such a way that visiting scholars are over there, do their research, contribute significantly the science. Thank you.
Thank you. So there were, okay. There is a question in the chat from Lawal, and the question is, what type of collaborations are available in BioRTC for cervical cancer research now? Mahmoud, would you like to take a stab at maybe answering what we could maybe do in BioRTC to address cervical cancer? What kind of methods maybe?
Sure. I think the main thing I would say is at the moment we have like the confocal, we have the qPCR, we have PCR machines, we have the Western blotting stuff, and the tissue culture biosafety cabinet incubators, they are on the way coming from Spain. So they should be available within, say, the next few weeks or maximum a month or two. So in addressing cancer, I think we need to be able to use cell culture to address those type of questions. So we would be able to perhaps, because I want to be very careful here, Professor Amadi made mention of one important point, that the collaboration should be driven by BioRTC's area of priority. Otherwise, we may end up doing so many different things at a time. So therefore, I would say we may be able to have that answer in the next few months when we have a clear idea of the type of research questions that could be addressed. But in terms of allowing other people to come in and use those resources, I think that is fine, because we said that we want to make it open. As long as those people collaborate, they get the training, or they are trained alumni, or they are through, let's say, International Society of Neurochemistry or IBRO funded programs, to conduct a research in a particular institution.
Great. Thank you, Mahmoud, for your answer. The next question, I feel as though we can answer in the chat, which might maybe work better with a wireless connection. So the question is, how can you help with building collaborations through and with BioRTC? What expertise can you bring in? And perhaps everybody who's on the call right now can type in the chat their expertise that they feel they could bring in and help foster collaborations with BioRTC. So feel free to enter in the chat different areas that you feel like you're coming from, different backgrounds, expertise. There's some interest in perhaps an immediate plan on creating immune suppressed mice or rats and specific brain disease models. We have also some expertise and interest in cancer cell gene editing. Okay, and perhaps we even have young students and doctoral students who would like to train their skills. And maybe would be looking at the BioRTC as a place to do so. I think BioRTC could serve as a great environment for doctoral training and student training. There is a question about which equipments are available for isolation of active compounds from plant extracts. Well, I think that we have basic biological equipment in the lab, like centrifuges, the ability to do extractions from plants, some wet lab equipment that would make this able to be done. Mahmoud, feel free to add in any potential equipment or ideas that maybe students might be interested in coming to BioRTC to learn about.
Yeah, I think the key thing to mention is, I understand that at the moment, a lot of people might be interested in doing their doctoral research. Some might be interested in postdoctoral or completing their MSc projects. And I think these are the kind of things that we can support. But again, I want to go back to what Professor Amadi mentioned. We have to be careful organizers to ensure that whatever is happening becomes, fits into the long-term strategy. So the best advice perhaps would be for people to look up the website to see what we have. And if they think as part of their doctoral or MSc or whatever project, they need some of those things that we have, they can then get in touch either through their supervisor directly, then engage us with their supervisors or institution to formalize collaborations. It's good that DVC is here, the registrar here, and all of us that are part of BioRTC are here. Then that way we can link them up within their own project. But also when we have the clear definition of the research project that we want to address, then people can also join our existing research projects, if you see what I mean. I think these are just a few ideas that people should be aware of. And there are a few ideas that we are discussing as well with Dr. Yunusa, Dr. Raouf, and other people about having a virtual institute where we have different collaborating institutions. But I think it is too early to mention that. But it's very exciting. People should look out for that.
Okay. Can I add something on top of? So yeah, definitely. I think, if you want to use BioRTC, immediately do check what equipment is available. But also, probably it would be useful for us, who is organizing the BioRTC, to know what is actually needed. For example, extracting some compounds from plant. We do have some, basic equipment for like biochemistry, but we do not have this more analytical equipment like HPLC or those stuff. And then if we know that there are actually quite high demand in this type of equipment, we could also try to find those equipment in somewhere else. And also the collaboration could be driven by what equipment is available, but also it doesn't have to be, this way, like, what is available, but also people just say, what actually you need for your own project. Then maybe like we could have actually some like a chart on the website and then saying that I want to do this particular experiment for my project. Is there anyone who can help, for example, in the local area, right? So that way, it doesn't have to be in the BioRTC, but we can connect people in the area.
Yeah, to quickly respond or to add to what Takeshi had just said, apart from the BioRTC, there are other laboratories, specialized laboratories, where we have the HPLCs. And in response to what the other person is saying, how to remove solvent from the plant extract, as well, they can use vacuum centrifuges. We have them in the universities and other laboratories. We also do have HVLC available in the university as well. So there are so many things that we can actually play around within the university. Or we can have an collaboration with other laboratories within the campus. Yeah. Thank you.
And to quickly just add again on what Takeshi said, which is very critical. If you do have something that we don't have on the website, you can still get in touch, because in a trend wants to make the BioRTC the center laboratory where the annual summer schools that Trend has been organizing for the past 11 years will be happening in BioRTC. So therefore we do have priority of getting equipment from other collaborators of Trend and ours, individually from across, the world. So if you come up and say you want to do this, and this is the kind of equipment that you may be interested in, even if we don't have it, we can then try to see how we can find them to ship them to BioRTC. As I speak to you right now, we do have freeze dryer in Sussex that is donated to BioRTC. Personally, I don't have any need for freeze dryer for any work that I'm interested in doing, but I still said, okay, I'm going to check it because I know that it may be useful for other people down there. So just get in touch. Let us know what you want, what you, we can then see how we can find it.
Hello, good day, everyone. Hello.
Hello.
Yes, good day. It's Hassan Sani. I'm from Federal University of Latvia here in Nigeria, and I have a few questions to ask. Can you hear me?
Go ahead
Hello?
with your questions. We can hear you.
Right, thank you. And right now I'm doing some work on the neurodegenerative diseases. I'm trying to look at gene expression levels of tau proteins, amyloid beta, and acetylcholine esterase. And I want to use Drosophila melanogaster as my model, but I have some few challenges. When I want to do the total RNA extraction, I don't know, will I use the whole head, or I will have to remove the brain, then do my total RNA extraction? So that is my question. Secondly, there is this issue of primers and kits. Is there a way that BioRTC can help me with those items? Primers for the tau protein and amyloid beta primer sequences, and also the acetylcholine esterase sequences. If I can get that from them, I'll be very grateful. And also, if I really have to even come down to their lab and get some of the works done there, I really appreciate. Thank you.
Thank you. Mahmoud, your hand is raised.
Could
no, I will lower it, but Rauf can answer that question regarding whether to use total fly head or just to isolate the brain. But in terms of the primers for tau and amyloid beta, so one of the key things that I should mention is we wanted to have a hands-on activity in BioRTC to formally launch it. In fact, this formal launch that we are having right now was supposed to be an on-site training, but due to COVID, we couldn't do it. So we are still planning to have this on-site training, most likely early 2022. And one of the areas of focus would be in neurodegeneration. We would come with some perhaps antibodies for tau, amyloid beta, ATC, but that will be for the purpose of training rather than for the purpose of this thing. The key thing for you to be aware of, if we have the equipment, there may be some consumables that are easily accessible, which we can give to you, but there will definitely be some consumables that you will need to purchase yourself, then come to the lab and use the equipment that we have to do the readout. So for example, if you want to do your ELISA, or if you want to do immunofluorescence microscopy, you still need to have your own antibody, then you have access to use the machines to study them.
Yes, to come back to the first question about whether using the total brain or the CNS. I think that the best way to do it is to use the CNS because of to be more precise. Because if we use the total brain, you will have, for example, a cuticle. You will have, for example, antenna inside. And as you know, cuticle themselves have their own RNA and their own proteins. So this may probably induce what we call a dilution effect. So I will suggest you use a dissect this the CNS of the Drosophila and then match the brain to extract the total mRNA from the CNS. I know that it's not easy, but I think that if you want to be more, you want to be accurate, it's the way of doing this.
Thank you so much.
Yeah, thank you. There was a question about how a PhD student enrolled at another Nigerian university can come to the BioRTC laboratory. In this case, I'm not exactly sure if I'm the expert about the different Nigerian universities and how easy it is to intercollaborate, but perhaps somebody on the ground there would like to answer that one.
Doc, do you want to respond to that? The DVC. The only thing that I will mention before I allow him to say is that if you the BioRTC is meant to be to work on the principles of open science. So in terms of collaboration, in terms of using the equipment, or other things like that, we would most certainly encourage that. But the only key thing is, which Doc will address, is whether how it will work in terms of, is it going to be joint supervision? Is it going to be that? How will BioRTC be acknowledged in that aspect? Because we are still developing this strategy and we have this long-term vision of having these virtual institutes that would share, would be joint supervising staff, but we haven't really written that down. But for now, let me, for immediate purpose, maybe Dr. Mohamed Musalon would say more about that.
Thank you, Mahmoud. I think in regard to BioRTC, I would say maybe we should wait a bit, but the university always do sign MOUs with other universities from across the globe in terms of sharing, having a co-supervision and so on and so forth. But I think with the BioRTC, I would suggest maybe they wait for now. Then after making all the formalities and seeing what the BioRTC can do, then we can come up with a statement on the website so that they can easily see what the BioRTC can go into. Yeah, thank you.
Excellent. Yeah, so perhaps it's 40 after right now. I think that maybe we should conclude this session. Perhaps I can just make a closing statement on behalf of the BioRTC and the Launch Symposium organizers. Just another thank you to all the speakers and the participants for being with us these past 2 days. I bet a lot of you are wondering now what's next. And that is an excellent question. We have learned over these past 2 days that biological sciences and neuroscience are integrative fields, encompassing a variety of methods to uncover difficult questions about how living organisms function. There has been clear expression of interest to collaborate and to continue interacting with one another on such problems. BioRTC is open to collaboration and invites all interested persons and students to get in touch. Please feel free to contact us on Twitter at BioRTCNIG or on our website, biortc.com. I put the link to our website in the chat. And yeah, basically the invitation is open to continue what we've started here with this launch this week. And yeah, that would be from my side. Thank you. Mahmoud, would you like to do a round of vote of thanks and add any further closing remarks?
Not really. The only thing that I would just emphasize is that we will be having a hands-on training to train people in BioRTC And once we have that hands-on training, almost everything will become much, much clearer, because these are expensive equipment that we have in BioRTC. And obviously what we want to do is to ensure whoever is using them has got the necessary training. We would discuss closely with the university as well as when COVID, once, because now COVID-19 restrictions are becoming much clearer, and advertise the call for this hands-on training. Perhaps at the beginning, we are only going to take maybe 20 people or 25 people. But once we're able to do that, then almost everything will become clearer. In terms of the last words, probably Dr. Maima Musalon should say the last words, even though, I'm a visiting associate professor there. Visiting is visiting, permanent is different. So Dr. Maima Musalon, I give it on to you, after which we can then close it.
Well, quickly, I can only say thank you on behalf of the university and the university community and the Vice Chancellor. We say thank you to all the participants, and we really need to sit with the team, with the BioRTC team, to see how we can work these things out, and to also call on the participant to be visiting the BioRTC website from time to time, so that any update regarding what is going to come up, they will find it there, actually. And thank you so much. Thank you, Dr. Mahmoud. And thank you, Reni, Takeshi, Sabi, and all that I'm not seeing on my screen. Thank you all so much for the time, and thank you so much for what you have done, the talks and everything. I can't thank you enough. That's it. Thank you.
Bye, everyone.
Bye.
Bye.
Congrats.
Yes.
Bye. See you again.